Hi, everybody. Welcome to another Chats with Children. I hope you're safe and well. I'm back in the UK from CPHI in Milano, and it's raining outside my window, but my day is going to be brightened because I'm here, delighted to be joined by Kelly Broster and Aaron Lamb from Thermo Fisher Scientific. And today we are going to be talking about, I've got to look this up, it's a bit of a long title, the topic we're going to talk about. We are going to be discussing the challenges around characterizing and controlling the development and manufacture of biotherapeutics. So that's the sort of overall topic we're talking about, and also dipping into and looking into MAM. So first of all, Aaron, Kelly, it's lovely to see you both. How are you?
Good, thank you. Yourself?
Yeah, well, I'm tired, but I'm happy to see you both. That's the thing.
Yeah, doing great, Rizwan. Great to see you again. I think
well, it's lovely.
Third time now we've done this.
Third time lucky, Aaron. Hope you will get it right this time. So yes, it's lovely to see you both. And hopefully it's sunny up north, but it's certainly not sunny down south today.
No, it's
well, I'm a northerner down south.
Oh, you're down south. So where are you based then, Kelly?
Just north of Oxford.
Oh, right, cool. Yeah, all right then. Well, that's good. The accent confused me a little bit. There you go. All right. So as always, before we start talking about MAM and characterization and all the other stuff, would one of you first of all give a very quick overview of Thermo, and then just give us a background on yourselves as well? So Aaron, do you want to kick off?
Yeah, sure. So Thermo Fisher Scientific is the world leader in serving science. So kind of a cliche, but what that means is like we support pharmaceutical development, healthcare, various other different industries. So across the plethora, we manufacture instrumentation for analytical science. We also have actual manufacturing of pharmaceuticals as well. So we really cover the whole picture, right? So my background, so I'm a vertical marketing manager for pharma and biopharma, but I'm an analytical chemist by training. So I've worked for Intertech Pharmaceutical Services at Manchester, and that was mainly on like small molecule characterization and control. But within Thermo, I've worked as like a senior application chemist. So I've got to grips with all the different types of instrumentation and method development across different markets. So that's my background. How about go over to Kelly?
Yeah, over to you, Kelly.
Yeah, so I'm in the same group as Aaron, but my background's more in systems oncology or cancer research. So I did a PhD in systems oncology and a postdoc for Cancer Research UK. I did have a past life as an application chemist as well, or a demo lab chemist. So I have used all these instruments before. But I've actually been in the role as a vertical marketing manager at Thermo Fisher in the biopharma team now for 6 years. So yeah, a bit about me.
Fantastic. All right, so let's talk about the challenges around characterizing and controlling the development and manufacture of biotherapeutics. So how has that process traditionally been carried out, and what are the challenges when you do those particular processes?
Yeah, sure. So obviously complete characterization of biotherapeutic proteins is a mandatory requirement for safe and efficacious drug production. And with protein-based therapeutics, any modification in the sequence, in the protein sequence or on the protein sequence can actually have a measurable effect on the drug safety or the biological effect of that drug.
Sure.
And therefore, regulators demand that we monitor that. And those modifications are termed critical quality attributes or CQAs. And what you have to do is you have to identify those, and then you have to monitor those all the way through development and QC testing.
Right.
And then traditionally, research or scientists use, lots of different analytical approaches,
right,
to measure these CQAs. So those can be, various different liquid chromatography based techniques. You can use, ELISA type approaches, some, capillary electrophoresis approaches. So the very diverse range of analytical approaches that people are currently using to measure and monitor the CQAs at the moment.
Right. Okay. And what are the challenges then when you are using so many different methods? Does that mean that there's difficulty sort of no consistency in terms of the benchmarks to measure the results, or what's the challenges around
it's
that?
very consistent because obviously these are regulated approaches, but obviously you need a lot of different, you need a lot of time. It's not streamlined. There's obviously cost associated with that. And some of the approaches in themselves don't really provide a depth of information.
Right. Okay. And Aaron, have you got anything to add to that?
Yeah, I think, I think Kelly's answered that really well. I think the major thing, and, if you talk about COVID and like the production of new vaccines and stuff like that, we really need to get drugs to market quicker. So, having multiple different methods, having multiple different analysts that need training in different techniques, having that spread across a laboratory, obviously it drains a vast amount of resources, right? It costs a lot of money, right? So if we can, if we can consolidate that down into, one system, one analyst, you can imagine the productivity gains, but also the cost savings as well. And ultimately, you're going to bring drugs to market faster. So, that's where the industry's headed, right? And, we can, when you're talking about analytical quality by design principles, that's kind of like, how we can consolidate methods down and make them, make them quicker. So that'd be what I'd have to add. So.
All right. Now I've heard the term MAM bounded about recently. So what is that? What does MAM even mean?
So MAM or MAM is multi-attribute method, or the multi-attribute method. And what does that mean? So really it's utilizing the power of mass spectrometry to measure all of these different attributes in a single analytical approach. So in a nutshell, that's what MAM is.
Right.
MAM.
Right. Now, I know that Thermo have launched MAM 2.0 recently. So why did you launch that? And what is actually MAM 2.0?
So I think if we take it back to MAM or MAM in itself, so obviously the industry is looking towards moving into adopting MAM. Many people have, there's lots of customers that are on that journey, there's lots of MAM pioneers, and we've been working with these for a long time, and the benefit of using MAM is obviously streamlining the analysis, but also using high resolution mass spectrometry, you're gaining increased knowledge and information on the process as well. So there's a lot of reasons why you would adopt MAM. It's very beneficial, like Aaron said, speeds up the whole development process. However, bringing in a completely new analytical approach that scientists are not used to using in those environments is not without its challenges. And this has been going on for a number of years now, and we've been partnering with various different customers, and we've been listening to them and listening to the challenges that they've had with the adoption of MAM. And we've been developing in the background, and we've got to the point now where we've decided to release, the next level MAM solution or workflow, which is what we've termed MAM 2.0. And what we've done is we've tried to, this is an ongoing process where we want to continuously work with our partners and customers to break down more barriers and help, the adoption of MAM in QC. But really with MAM 2.0, the benefits that we've now got a Orbitrap-based mass spectrometer that's purpose-built for a QC environment. So that's great, but that's also supported by a software ecosystem which can support the whole journey from development into QC. And we've got seamless method transfer. So all of your methods that you're creating in development, we can transfer them in a compliant ready environment over to QC. So these are the types of things that people have struggled with when adopting MAM in the past. And we've just trying to look at the main hurdles and help our customers break them down as we go. So right. That's what MAM 2.0 is about, really. It's years of listening to customers and the result of our learning from that.
So would I be right in saying is really more of an integrated solution then? Because that's the buzzword I've been hearing at CPHI all week with different exhibitors in every single hall, no matter what they were talking about in which hall. It's all about integration and making sure that you can
seamless platform, really, all the way from development into QC.
Right. Okay. So, Aaron, have you got anything to add to that?
Yeah, I'd just add, that linking the development to the QC is the critical part. So, we all know that MAM is the next step forward with the characterization of biotherapeutics, because it simplifies down what is could be 10 different methods on 10 different instruments into one platform, which is an LCMS based approach. But having that link between, development and QC, so development is all about finding out what the principal quality attributes are, what are the peaks that are produced, or the products that are degraded or deamidation, the various different post-translational modifications that can cause problems with the production of the biotherapeutic. So identifying those easily, so new peak detection and stuff like that, is something that we've built into the MAM 2.0 platform. So it's all about the software side of stuff mainly, and having the high resolution mass spec as well allows you to confidently identify these. So this is a key advantage of the development side, but having that compliance link between the development and QC is critical. So being able to transfer that CQA workbook over to QC so that you can monitor the compounds of interest is something that is seamless now, and it's controlled in a GMP way as well. So that's all controlled through the software additions that we've had. So that would be my main point to add.
So just as a layperson listening to this, and you talk about mass specs, does that mean that the other methods that were traditionally used are sort of now not needed any longer, or are they still part of the overall process?
That's a difficult question right now, because I think there's going to be a period where people are going to be using both and validating the two, but it is a movement across. So at the moment, people are still very much using traditional approaches, and if they are adopting new approaches, they'll be using them together initially. And then it depends where you are on that journey to adopting MAM based on what you're doing. But for a while, it'll be the two. It's not something that you can just pick up and put down. It's a journey.
Right. Okay. So when did you actually launch MAM 2.0? And how can people find out more about it? Aaron?
Sorry.
The beauties of doing
the background. Perfect timing.
No, well, look, that's the wonders of doing interviews over Zoom. Don't worry, mate.
No, so we launched it at ASMS. So that was actually last week.
It was, yeah. I wish I was there, but I was
there.
I was in Barcelona instead. So there you go.
Well, that's equally as nice a city, right? Or better, probably. So we launched it at ASMS. So we had breakfast workshop. We had some giveaways as well. So we created a Lego version of the new Orbitrap Exploris MX, which is used in the QC lab. There's also a MAM Rubik's Cube that we created as well. So you can check out some of the posts that we'll drop in the chat below. But so we're really taking it forward with a virtual event that we've got upcoming as well. So
right.
If you want, I can share.
Yeah, please do. But before you do that, when is the virtual event?
The virtual event is on the 18th of November.
Right. Okay. So next week. Yeah,
we're creating so I think everyone's bored of death by PowerPoint, right?
Yeah.
And traditional webinars, we've seen tails off in people attending and stuff like that. People want to really get out and see the workflows in person. So ASMS, we had a big booth, we had the workflows, we had software demonstrations where you could come up and see the workflow from development right through to QC. But obviously with travel restrictions, not everyone could attend ASMS. So we will be doing a series of road shows next year. So we'll be doing in-person events. But with the online event for everyone, we've created a virtual lab environment. So we've actually created a QC lab and a development lab, and within each of these labs, there's a virtual lab tour. So you can see an overview of the hardware and the software, and then we also go into the actual workflow from sample preparation, so introducing the sample into the system, to getting a result at the end, right, on both the development and the QC side. So I'll share my screen if you want. and I'll
All
show
right.
you that.
And fingers crossed with the wonders of technology this works. So let's see.
Yeah. Okay, can you see my screen?
I can, yeah.
Okay, so this is the virtual lab environment. So this is the development lab. So this is where we're doing like the research and development of the new biotherapeutic. And we can see a nice little background here. I wish I was
In San
working in this
Francisco.
lab.
Yeah, there you go.
Yeah.
Yeah.
Yeah, so Melissa Sato, who's one of our key like MAM experts, along with Royston, Quinton, and Kai Scheffler, and also Mark Yang. These are the pioneers from Thermo in the development of this MAM 2.0 solution. So what we can see is we've created this virtual lab environment. So this is the HPLC system. So this is the Vanquish Flex.
Sure.
And then this is the Orbitrap Explorers 240. So this is the mass spectrometer. This is what does the detection of the peptide fragments. So this essentially is a peptide mapping workflow. And within the lab, so there's going to be like an overview of the hardware system by Kai Scheffler. And then that's going to lead into the software demonstration. So the actual, how do you set up this system? How do you, acquire data? How do you process the data? How do you create the PQA list, which is the principal quality attribute? How do we then go on to create the critical quality attribute list? So the critical quality attributes are from the peptide. Once you break it down into its individual subpeptides, there's only certain ones that are critical to the control of the actual production of the biotherapeutic.
Yeah.
And then once you've identified those, usually through toxicology risk assessments and such, you then send that list. So what we call it is an e-workflow. We have a list of peptides that we have to monitor, these CQAs. We send that to the QC lab, and then basically you're just monitoring in a targeted way. So the development side of the workflow is very much like a discovery sort of phase.
Yep.
And then once you've really nailed down that list, okay, what are the key components of this biotherapeutic peptides that are critical to the control of it? So we then send that through to the QC lab, and the guys that are going to give these software demos will really take you through, and you will really understand completely exactly how this workflow works. So this animation here is the breakthrough. So like one of the key aspects of this workflow is we want to accelerate your biotherapeutic straight through to breakthrough. So there's going to be a little animation there, and then we're going to lead into the QC lab. So I don't know if it's going to load because we had some technical issues before, but we'll give it a go. And
it does. There you go. Great sigh of relief from you, Aaron. There you go. You feel better there. So
in the background, I don't know if you can make it out, but,
oh, it's Dublin there, right? Okay, cool.
You can see it. Yeah, you got it.
See, we shouldn't have told anybody this and done a little quiz at the bottom of the video saying guess where we were. So that would have been even better. So there you go.
Yeah. So this is the QC lab, and again, this is going more into the this is a quality control where we've got a targeted list of compounds that we're looking for, and we've got reporting templates built out, and basically you just want to see if it's passing or failing, right? So you want to make it as simple as possible, and it is. And we can see we've got lots of systems here. So this is a very futuristic MAM lab. Yeah, we're really looking forward to hosting people in the event.
Brilliant.
I'm going to stop sharing now. Hopefully that was a good overview for you.
No, that's great. So is there anything else you'd like to add, though, about MAM in general or MAM 2.0?
I guess from my side, we just want to see what the customers see what they can do with this now and see how much this helps with their adoption, because the power's there. We know that it's going to make a significant difference in how quickly drugs can get to market,
right?
And obviously that's what everybody wants. That's what we're all here for. So yeah, we're very excited that it's out there, and looking forward to kind of looking at what our customers do with it. That's the main thing.
Brilliant. And Aaron, anything else to add? before we finish up?
I'd just add, so like this, we've got a new web page as well. So like there's lots of information around MAM 2.0 and MAM in general. So because, we really want to take people on a journey from like, okay, there's a lot of new adopters to this technology. We know it's not, broadly established. So we understand we've got to take people from different aspects. There's the pioneers that obviously know MAM really well, but then people that are getting used to it, we need to cater for that. So this virtual event will do that. So there's a learning center, which will take you up to scratch. What is MAM all about, right? So we've got some videos, we've got some different materials that people can consume, and I'd encourage people to come to this platform. And after the live event, this platform is going to be available on demand. So there's going to be a lot of information that people can consume that's not part of the actual the live event. Really, really excited to get people engaged in MAM, because we do think it is the future, and it is all about accelerating biotherapeutics to market.
Fantastic. Well, look, thank you both for taking the time to talk to me. Thanks, Kelly. Thanks, Aaron. It's lovely to see you again, mate. And I hope the pipe gets fixed very quickly.
And me too. It was leaking all over the floor.
Well, you were very calm in doing this interview, so I'd have been stressing out about that rather than doing the interview. It has to be said. So, good luck with the launch. I hope it goes really well. Good luck with the event. There you go, viewers. If you would like to know more about MAM 2.0, I can't really say MAM. I feel a bit bizarre saying it, but MAM or MAM 2.0, then check out the link below so you can register for the event on the 18th of November. It is free, and if you register and you can't make it, you can obviously see it on demand as well. So all the links will be below I say below, it's actually above the video, so that you can click on those links. And if you've got any questions for Aaron or Kelly about what they talked about today, then feel free to put them in the comments below the video, and they'll be sure to answer them, I'm sure. And you can also connect with them on LinkedIn. They're both on LinkedIn, and you can send them messages via that as well if you want to. So all that's left to say, as always, is Aaron, Kelly, thank you very much. It's lovely to see you.
Thank you.
And hopefully we'll catch up in person in the not too distant future. And viewers, I hope you found that useful. As I say, if you've got any questions for Aaron and Kelly, please leave them below the video. Register for the event on the 18th of November. The link is above the video, and it's all free, and you can learn more about MAM in general and MAM 2.0. And until next time, as always, hopefully the sun will shine for you over the weekend. Stay well and stay safe. Bye-bye.
Take care. Bye-bye.
Thank you.